chemicals for water filtration systems. Diatoms are probably even in your toothpaste.
For its contributions to evolution, the food chain, and dental hygiene everywhere, we salute the diatom, a shining example of a micro-hero!
And what's this ghostly specter riding the waves? Another seafaring protus known as a dinoflagellate. Like fireflies, some dinoflagellates glow, lending shimmer to the nighttime seas.
Attention beach lovers. If you want to swim in the human race, you've got to watch out for the microphobes.
It's a me!
They may be flashy, but some dinoflagellates are parasites, organisms that feed on other living things.
That's a symbiotic relationship that benefits only the parasite.
When they get together with a few billion of their close relatives, it's no party. It's a disaster. One type of dinoflagellate exposure is known as a
Where's the party?
Sometimes covering several hundred square kilometers of water, it poisons marine life and can cause all sorts of illnesses.
So many visitors.
Oh, and did I mention that a red tide really stinks? As the leading cause of a rotten day at the beach, these dinoflagellates get our nod as microphobes.
Although all the protists we've discussed so far can use photosynthesis to make their own food, there are many more that cannot. Like us, they depend on existing food to survive. Some of these organisms appeared so animal-like to scientists that initially they called them protozoa.
Proto is Greek for first, and zoa means animal. First animal. Get it? Except now they're not animals at all. They're protists.
Well, you can see why people used to think they were primitive animals. As we mentioned before, they're heterotrophs. Like animals, they get their food from an outside source. Also like animals, these protists have no cell walls, just a soft cell membrane. And last but not least, most move under their own power, just like
Many protists have developed some amazing ways to get around. For instance, the amoeba makes full use of being a softie. It simply extends that soft cell membrane out like a hook, surrounds its target, and swallows it up. Other protists propel themselves through their environment with hook-like tails known as flagella. But why have one propeller when you could have hundreds? Tons of tiny hairs cover the protists known as ciliates. Each hair is a cilia, a Latin word that means eyelash. Looks sort of like they're batting their eyelashes, doesn't it? But really, they're beating their cilia so they can get from here to there.
The well-known ciliate, paramecium, is one of the most complex single-celled organisms of all. It can usually be found in any freshwater pond. Darting to and fro, it detects and hunts its prey. It has no real vision, yet it senses and avoids anything obstructing its path. A paramecium reproduces in a way that is typical of most protists, binary fission.
Binary means two, as in the number 2. Fission means to divide. So, binary fission is a fancy way of saying split in two.
When conditions are right, a paramecium will divide 2 or 3 times a day. With that kind of growth, it's a small wonder that just 1 liter of pond water can hold millions of those critters.
Not all protists hang out in ponds. Some of them are more intimate creatures, like the cozy insides of other larger creatures. Creatures like you.
Some are welcome guests. They help break down the food that would otherwise be indigestible. But others are very nasty characters indeed. When the African tsetse fly bites, it transmits a protist which causes African sleeping sickness. And Plasmodium is the protist behind one of the world's most feared diseases, malaria. Malaria presents a huge health hazard to people who live in and around water in the tropics.
Now, who figured that out? A science sleuth, of course.
In the 20th century alone, more soldiers suffered and died from malarious fevers and chills than from bullet wounds. So it's not surprising that it took two military doctors to unravel the mystery of malaria. The first was French physician Charles Laveran. Looking through a microscope in 1880, he spotted plasmodium wreaking havoc in a patient's blood sample. He knew he found the enemy, but how had it gotten there? It took 9 years and a second sleuth, Sir Ronald Ross, to put that piece in the puzzle. On an expedition to Africa, Ross proved that the parasite was hitching a ride with mosquitoes. For their important part in fighting the battle of the malaria bug, both Dr. Laveran and Sir Ronald were awarded Nobel Prizes, and we give each of them the honorary title of Science Sleuth.
Okay, you guys. So you got all this Crota stuff figured out? And you'd be able to identify their characteristics if you saw one? Well, then let's try an experiment and see.
I've got a good purpose to test. Euglena.
First, you need a concentrated culture, which is science speak for a condensed batch, of euglena, a microscope, a petri dish and some aluminum foil, a sunny window, and, of course, a notebook and pencil. Now we'll make a slide of euglena and see what we can see under the microscope.
Right off the bat, we notice that euglena is green. That's a dead giveaway, and it's chlorophyll, a key element of photosynthesis. So perhaps you'll say it's plant-like. But hold on here. These babies are moving around. That makes it seem more like an animal than a plant. They even have a spot that looks kind of like an eye. What do you suppose that's for?
That's what we're going to find out. Give the culture a swirl to get the organisms uniformly dispersed in the culture. Then pour the euglena concentrate into the petri dish, enough to cover the bottom evenly. Take a good look at the culture and write down what you see.
You should see little green particles floating around.
Okay, now let's cover it up and bring our little friends and the aluminum foil over to the window to catch some rays. Set the petri dish carefully down on the windowsill and then cover half the petri dish with the foil.
Now we'll expose these protists to light for about an hour. But while we're away, think about your observations of the euglena. They're green, they can move, and they have that mysterious spot. Can you predict how the euglena will react to the light?
Okay, time's up. Let's check back in with our little green friends. Maestro, remove the foil, please. Aha! It appears that euglena is not content to sit in the dark. In fact, most have moved to the uncovered half of the dish, into the light. Any ideas why? Well, first of all is the green color you observe. Euglena does contain chlorophyll, enabling it to produce its own food through photosynthesis. And to do so, it needs light.
That's a very plant-like quality, but plants usually stay in one place. Euglena pursues its energy source, more like an animal. And that spot we observe, it's actually called an eye spot. It detects light so the euglena can travel toward it. In conclusion, we can say that while euglena is a protist, it shares several traits with other types of organisms, namely plants and animals.
I can see why scientists have had such a tough time classifying protists.
The most baffling protist of all have to be the ones that seem to be like fungi.
Fungi! Those are the organisms like molds and mushrooms. They've got cell walls, but they don't move around much. And, like all good decomposers, they break down stuff that used to be alive. How could a protist be mistaken for a fungus?
The most notorious example is the protist that produced the Irish potato famine. This 19th century crop disaster caused more than 1 million deaths from starvation in Ireland. Generations and the work of a fungus. But thanks to more recent advances in cellular biology, scientists identified the culprit as a protist.
And now, simple organisms present the secret life of slime molds. When last we saw our mystery slime mold, it was hiding in the daily process. Was it a true mold? In other words, a fungus, as its name would suggest? Or was it really just a very jittery single cell able to travel like an amoeba? Or was it perhaps the star of a horror movie featuring alien blobs?
Slime seem to be manifestos with personality. When you're under stress, you can form spores and bodies, kind of like mushrooms. In fact, slime molds look a good deal like fungi.
Oh, so I'm a fungus, right?
Well, you do decompose dead organic matter and devour any unfortunate bacteria that cross your path. But nobody who sees you in your juvenile state could ever mistake you for a fungus. You don't have cell walls, and you live on the way no self-respecting fungi will ever do.
Oh, so I'm an animal, right?
Wow!
Soft-cell animal, able to move around to find my lunch.
Wonderful!
Most certainly not an animal. No animal was ever able to do this. Unite thousands of independent cells to form one big, single, slimy, multi-cell organism. It's like a creature out of a bad science fiction.
Will the true identity of this ever-changing protist be revealed? Tune in next time when simple organisms present the secret life of slime molds.
The slime molds are just one more fascinating facet of the most varied kingdom of science, the kingdom of proteins. They join such amazing creatures as glow-in-the-dark dinoflagellates and the glass-encased diatoms.
Some protists, like the paramecium, can move about and hunt prey like animals. Others, like Euglena, act an awful lot like plants. And the slime mold looks so much like a fungus, scientists actually call it mold. But are they really plants, animals, or fungi? If the answer's not quite, they're protists.
Paramecium, Euglena, and slime molds. Protists demonstrate plenty of different eating habits. But they do have a few things in common. They usually reproduce through binary fission. They all have at least one nucleus, and many are single-celled and microscopic.
But others are large and multi-celled.
It's true that it's often easier to say what a protist is not than to say what it is. That's why these simple organisms seem so complex. For protists belong to the kingdom that refuses to be defined.
I was curious how much it's hard.
Oh, yeah!
All right, we've got about 5 minutes here, so answer one more question and then we'll look at